Assalamualaikum to everyone, and this is Miss Hanis. I'm going to teach you on fermentation. Okay, so I can't really speak loudly because it's nighttime. Okay, so please turn up your volume if it's too slow to you. I'm trying to talk as clearly as I can to my mic, and I hope you can listen to me. All right, so look at the pictures here. We have some bread. Okay, very good looking bread here, very tasty. And this is croissant. And I actually put the croissant here not because it uses some yeast. If you know, to make croissant, we don't really need yeast. But for bread, okay, this kind of bread here, can you see it? This kind of bread and this one, we actually need yeast. Okay, because why? We need to rise the dough, or else the dough will not rise. Okay, so why are we learning about fermentation in this lecture? So you have to remember the title of this topic is cellular respiration. Okay, cellular respiration can occur with and or without oxygen. So the one that we have learned so far for the subtopic 5.1, okay, the one that we learned about glycolysis, pyruvate oxidation, Krebs cycle, and finally oxidative phosphorylation, which consists of electron transport chain and chemiosmosis. All of those are under aerobic respiration. But fermentation, it can occur even without oxygen. Okay, so for this lecture, we're going to focus on cellular respiration that needs no oxygen or lack of oxygen. Okay, so here are the learning outcome that you need to know. Fermentation and its application. So mostly, okay, we learn the first part is learning about the lactate and alcohol fermentation. I believe that you guys have learned this back in SPM. So what is the difference between producing lactate and also alcohol? We will see later, okay? We'll try to flashback of what you have learned before. Okay, and B, the importance of fermentation industry. So there are 3 kinds of industries that we are going to focus on. Of course, there are a lot of it, okay? But for this semester, okay, for your level, we're going to focus on bakery, okay, using yeast to make bread, or vinegar, beverage and alcohol production. As you guys may know, we use grapes and then we ferment it for a long time to make a delicious wine, okay, for alcohol, and also fermented food, which are actually very good for our health. Okay, some examples include the tapai, kimchi, and tempoyak. I honestly don't like any of this. I like bread. Bread is good, but not tapai, not kimchi, and tempoyak. But if you like it, then you're gonna enjoy this topic. Okay, so let's see the definition of fermentation. What is fermentation? So you guys have to know what is fermentation. Fermentation is a way of harvesting chemical energy without using either oxygen or any electron transport chain. You have to remember, when it comes to ETC, we need oxygen. Why do we need oxygen? Okay, oxygen is O2. Why? Because just remember, oxygen acts as the final electron acceptor. Okay, final electron acceptor in oxidative phosphorylation. Okay, so that's why in fermentation we don't need any oxygen or ETC. And it is an incomplete oxidation of glucose. We know that the glucose is oxidized, but in aerobic respiration it is complete. Okay, complete. But in fermentation it is incomplete. Okay, so pardon my bad handwriting here. I'm trying to write using my mouse, okay? And you have to remember, when it comes to incomplete oxidation, we, okay, produce CO2, ATP, and ethanol. But this one, alcohol, is just in plant cell. How about animal cell? Like us human, we produce lactate and ATP. Look at this. And it's been underlined. So if you don't put all of this, okay, for example, in your exam later, PSPM semester 2, if you don't put all of this, maybe you just remember, okay, I'm just gonna put ethanol. Then that is wrong, because you have to put all products, okay? And then this is very important too, in the absence of oxygen, because it needs no oxygen at all. All right, so let's take a look at fermentation. You have to remember, when it comes to fermentation, just like glycolysis, it only produces a small amount of energy. It's not a lot, okay? It's just like a very, very little. Okay, it only generates ATP by substrate level phosphorylation. Why? Please remember, there are three ways. Okay, there are three ways to generate ATP, and the first one is substrate level phosphorylation. The second one is, what is the second one? Oxidative phosphorylation. But remember, oxidative phosphorylation is included in what? After Krebs cycle, right? Okay, oxidative phosphorylation. It's taking me a long time to write this down. My bad. Okay, so oxidative phosphorylation only happen after Krebs cycle and need oxygen. So that's why they say here, only by substrate level phosphorylation. And the third way to generate ATP is by using photophosphorylation, which is actually covered in chapter 6. So no need to know about that yet. We will cover about that later on. But please, if you still cannot differentiate between these three ways of generating ATP, please watch again the first lecture that I've sent you before. Alright, so look at this. Fermentation consists only, okay, by glycolysis. Because why? I keep telling you guys, okay, in class, glycolysis do not require oxygen. That is the best part of glycolysis. Even without oxygen, okay, glycolysis could occur. If you take a look at here, this is glycolysis. I hope everyone is familiar with it. You guys know glucose, 6 carbon, and then it will be separated into 3 carbon. But it's not like, okay, just separate, okay, done, we get 2 pyruvates. No. Okay, it consists of series of, look at this, we have like intermediate substrate. So that is why in the end we get 2 pyruvates, which each one of the pyruvate consists of 3 carbon. Okay, so please remember, fermentation only consists of glycolysis. That is the best, but even though it just generate like 2 ATP, okay, just 2 ATP in glycolysis, but no need oxygen. Alright, is that clear? Okay, so let's take a look a little bit on fermentation. So glucose to 2 pyruvate. Okay, so of course, when we oxidize glucose to 2 pyruvate by glycolysis, okay, we get 2 ATP. Please remember, we invest 2 ATP in the investment phase, and then we get back another 4 ATP in the payoff. But before that, we invest 2 ATP, right? So 4 minus 2. So net ATP is 2 ATP. Okay, and also in glycolysis, we actually produce 2 NADH. That's why they put it here, 2 NADH plus H plus. It is completely okay if you just say, okay, produce 2 NADH. But if you want to say the full one, 2 NADH plus H plus, that is fine too. Right? So this is how glycolysis works. Okay, everyone knows about this. Hopefully, right? Okay, I believe in you guys. And lactate fermentation. Okay, so now we are focusing on animal cell. Look at this picture. Okay, these are, I believe they are Japanese. Okay, so there are some Japanese people, okay, doing marathon here, running. Have you ever run before? So if you're a beginner to running, it actually takes very, very short time for your muscles to feel like, oh, it's so tiring, you know, like cramp. You got cramp in your muscle because you know what happened there. Okay, so when we exercise, the oxygen level, okay, because we actually respire a lot during exercise, like, okay, you're panting, you need more oxygen, that's why. So now the oxygen level is decreasing. Okay, the oxygen level decreases. So if there is not enough oxygen, what happened? So your muscle will undergo fermentation, or more specific, please use lactate fermentation. Okay, so that we know this actually happened in animal cell. So what happened here, if you just run out of nowhere, okay, our muscle cell is depleted of oxygen, not enough oxygen. So that's why we will produce lactate. Okay, let's take a look at this. So please remember, after glycolysis, after we oxidize the glucose, we get pyruvate. Okay, glycolysis, 1 glucose, we get 2 pyruvate. Okay, 2 pyruvate, 2 pyruvate, and 2 NADH. Okay, and 2 ATP. Okay, everyone knows about this already. Okay, and then remember, if we don't get enough oxygen, what will happen to the pyruvate? Do you think the pyruvate will go oxidation? Okay, pyruvate oxidation? Nope. It will be reduced instead. Okay, so now it's vice versa. It will be reduced by NADH to produce lactate. So it's another step. Okay, you have to remember this is like another step so that we get enough ATP. That's why our body does this. Okay, so when NADH is used again, of course, it will be converted back to NAD+. So why? So that glycolysis will use more NAD+ to regenerate NADH. So it's just like a cycle. Okay, so let me just put it, yeah, this is just like a cycle. NAD+ to NADH, NADH to NAD+. Okay? So look at this. When it comes to glycolysis, so glucose, actually, this one is not really complete. You can put here like glucose to, yes, pyruvate. Okay, and then the pyruvate will be reduced to lactate. Okay, so you can put here pyruvate. Make sure to add another arrow so you can see glucose to pyruvate. So we all know pyruvate oxidation is just like the link reaction. Okay, the link reaction to Krebs cycle. But now, no oxygen, so no pyruvate oxidation. Oxidation need O2. So no O2, no pyruvate oxidation now. Okay, so let's take a look at the next slide. So this is how it looks like. From pyruvate to acetyl-CoA. But this one, if oxygen is available. If O2 is available, then the pyruvate can undergo pyruvate oxidation. If O2 is available. If not, it will undergo fermentation. Okay, lactate fermentation for animal cell. Is that clear? So look at this. Glucose to 2-pyruvate. Now, instead of being oxidized again, no, it will be reversed. Okay, it will be reduced. So that's why the arrow is like this, U-turn kan. U-turn. So this one is oxidation. We know that the glucose is oxidized. Oxidized. I'm going to put it here, oxidized. And the pyruvate will be, instead of being oxidized again, just like the name, pyruvate oxidation, nope, now it's being reduced. So if it's reduced, of course, the NADH also will become NAD+. And that's how we get lactate. Look at the extra information here. Brain cell does not undergo lactate fermentation according to the Campbell textbook. So let me know why in the comment below. Why do you think brain cell does not undergo lactate fermentation? Like, what's the matter? Why not brain? Okay, so try to find the answer for me. And let's see. This is how it looks like. You keep massaging your muscle. Okay, because it hurts. It's starting to hurt. Okay, so why do you think it causes muscle cramp? Because too much lactic acid. So lactic acid is the same as lactate. Okay, so when we're talking about lactic acid, it's the same thing as lactate. So how do we actually restore the muscle to the normal state? Okay, so the lactic acid is very, very toxic in our body. It cannot stay in our body or else we might die because it's too toxic. So what happened? Okay, this is our muscle here. The glucose is converted to lactate, right? So the lactate actually transferred to the liver. Okay, our liver is very amazing. The lactate will actually undergo gluconeogenesis so that we can produce glucose. Okay, but this one needs some oxygen. So it's called Cori cycle. We don't learn this, don't worry. It's just extra information. But you have to know that the lactate is transported to the liver. Okay, so that it can regenerate glucose for muscle cell. Alright, is that clear? This one is very toxic in our muscle, so it will be transfer to the liver. This is the liver, so that we can generate more glucose. So this is a cycle here, as you can see. Okay, so enough on lactate fermentation. Let's talk about alcohol fermentation. So just now it occurs in animal cells. So of course animal cell, after that is plant cell. Okay, or you can say yeast. So when oxygen is insufficient, again, glycolysis occurs to oxidize glucose into pyruvate. But after that, do you think pyruvate oxidation will happen? No, because no O2. So that is why instead the pyruvate is converted into acetaldehyde ethanol or ethanol through decarboxylation. And then again, acetaldehyde is reduced by NADH to produce ethanol. Okay, this one wrong spelling, ethanol. I know everyone is so good in chemistry, so probably you have noticed about this. Okay, ethanol, alcohol. So let's take a look at the picture right away. For lactate fermentation, it's just one step, right? From pyruvate and then we get lactate. But for alcohol fermentation, we have another one extra step. From pyruvate, it will be converted to acetaldehyde. How? Look, the CO2 is now released. So if CO2 is released, the process is called as decarboxylation. Okay, carbo kan, carbon group. Not carbon group, I think it's carboxyl group. My bad, I'm not good in chemistry. Okay, so decarboxylation. Okay, decarboxylation. Okay, carboxyl group, we release CO2. So that's why we call decar. Okay, like decaf, no caffeine. So no carbon dioxide, decarboxylation. Okay, so please remember we have like 2 steps here instead of 1. Okay, from pyruvate to acetaldehyde and then acetaldehyde finally to ethanol, which is alcohol. Ethanol is alcohol. Okay, this is something that you have to take note. Alright, so you have to know the difference and also the similarities between lactate fermentation and alcohol fermentation. Okay, so I'm not going to touch more about this. You can read it on your own. It is very easy. So let's move on to the importance of fermentation in industry. So in bakery, okay, of course, to make this bread, very, very yummy bread, we have to use yeast. Why yeast? Have you ever thought of it? Like, why do we use yeast? Because in yeast, actually, we have an enzyme. Look at this, ASE. So this is an enzyme called zymes. Okay, so what zymes actually do is, the starch, we actually used wheat flour. Or you can use any other flour. So wheat flour has starch in it. So which flour will be hydrolyzed to maltose thanks to zymes. Okay, and then you have to know, without the presence of O2, must no need O2, O2 cannot be present. Okay, only no presence of O2, the yeast can undergo alcohol fermentation. If you ever make a bread, right, you have to close the dough or else it won't rise. Because you know why we have to close it? So that the oxygen will not enter into our dough. Okay, so when we close the dough, it will double the size like what you can see here. And that's how you know the yeast is working. So why does it double in size? Because the CO2 is released. That's how you know. Okay, so why do we actually use this in our bread? Because it actually gives a very unique flavor in bread. Okay, so please remember all of these key points. So this is about bread. Very nice. And the second one on vinegar, beverages, and alcohol production. So again, we use yeast. Yeast is very good. Okay. Oh, just some fun fact on yeast. Before this, okay, I don't remember in which era, but before, humans thought yeast is dangerous because they feel like, oh, look at it. It is actually alive. So around some time, okay, we actually stopped consuming yeast. But we started consuming yeast again once it is proved to be safe. Okay, so imagine a life without yeast, no bread, no vinegar, and no alcohol for some of you. Okay, so look at this. How do we actually produce vinegar and alcohol? So the yeast can actually feed on the sugar, right? Such as fruits, whole grain, potatoes, or rice. Like a lot of options here. Okay, so all of this will be turned into liquid. Okay, this, we don't just use fruit. We have to mash it. We have to mash the potato or rice until we get the liquid. So the liquid will be combined with yeast so that it can be ferment into alcohol. Okay, and then just after that, the alcohol is exposed again to oxygen. And then we add another bacteria called acetobacter. So please take note of this, acetobacter. Why? Because we want to ferment again. So for weeks or months. So sometimes for years, right? Okay, so the longer it is, the better it will be. And the more expensive it will be. Okay, so this is how they produce alcohol, vinegar, and also any other beverages. Okay, so actually I have a video here. I'm not sure if I can play it. Let me try to play it. I don't think I can do it. It's okay. I will give it to you on Telegram group, or you can just search it, okay, on YouTube Shorts or TikTok. Like, how do they actually make wine? Okay, so that's how you can actually visualize this process. Okay, so, and look at this. Now we have tapai. Okay, tapai. Also, we use yeast. Okay, we use yeast. And then we actually, you know how tapai is made? Okay, you can look at this here. I think they use cassava here. Okay, cassava. And tapai. Tapai, you know how to make tapai? They just put some yeast and then let it stay there for days and then done. Okay, so you may watch this YouTube shorts. I can't play it. It's so sad I cannot play it for some reason. Okay, and then now we have tempoyak. Same thing. Okay, but for tempoyak, usually we put some salt or sugar. And then we keep it at room temperature and left it to ferment for 3 to 5 days. And you have to remember, we also have this bacteria called lactic acid bacteria. Remember lactic acid? Okay, the bacteria that produce lactic acid. Oh, they actually has another name, which is lab. Okay, so when you see this lab, not this lab, but LAB, it must be in capital letter, LAB. So lab can actually be introduced to the fruits, or it's actually occur naturally as well. So it can be both naturally occurred or being introduced by us. Okay, so lab is very important because this actually will inhibit the bacterial growth. So that's why it's good. Even though you just keep it at room temperature, it will stay good. You can eat it. Okay, so this is tempoyak. I don't like tempoyak, so let's move on. Okay, and finally, one of the most popular food nowadays is kimchi. So again, kimchi is a traditional Korean food. And look at this. They actually introduce the lab. Okay, why? So that it will last longer. Right? So the bacteria actually metabolize. Okay, they eat the sugar. The bacteria eat the sugar so that it can be turned into lactic acid. Okay, and then you know the lactic acid, even the name suggests acid, so it's sour and usually increases the acidity. So it's actually safe to eat because you know why? Why is it safe to eat? Usually the bad bacteria like E. coli cannot live in extreme pH. So acidity is actually quite extreme, so they cannot live there. Alright, so this is actually the last slide for fermentation. I hope you guys enjoy it. If you have any question about this, please let me know. Alright, so these are all you need to know. Please remember some of the keywords. It's actually quite interesting topic to cover. And I'll see you later. Okay, thank you for listening and watching. Bye bye, take care.